This video outlines the NMPA 2025 registration roadmap for oral repair membranes, which are Class 3 guided bone regeneration devices in China. We will cover the strategic pathways and compliance architecture necessary for successful registration. Oral repair membranes create a physical boundary, prioritizing space for slow-growing osteoblasts in guided bone regeneration. This function blocks soft tissue migration, secures the spatial void, and facilitates osteoblast proliferation for bone growth. Oral repair membranes fall under Class 3 medical devices, the highest risk category within the NMPA classification. The generic naming convention requires aligning terminology with the medical device generic naming rules, as shown in the compliant examples. Registration Unit Division uses bifurcation rules based on 4 gates: material, biological source, processing method, and dimensions. Variations in raw material, biological origin, or core preparation methods mandate separate registration dossiers. Size variations do not. Structural integrity and physical thresholds are evaluated through macroscopic forces like tensile strength and microscopic architecture like porosity. Environmental reactivity, such as water absorption, must also be tested to ensure clinical fit. Chemical performance evaluation standards differ for collagen-based and silk fibroin membranes. Both share baseline requirements, while specific assays are required for each material to validate composition and characteristics. The degradation versus regeneration timeline is critical. The membrane's structural integrity must be maintained until sufficient bone tissue regeneration has occurred, establishing a critical barrier window. In vitro testing mandates simulating the enzymatic environment and tracking performance. The animal origin biosafety defense architecture focuses on 3 layers: source control, pathogen inactivation, and immunogenicity elimination. Success is measured through residual cell counts, DNA quantification, and alpha-gal antigen levels. Comprehensive biological evaluation follows ISO 10993 and YYTO 268 standards, covering 8 areas, including cytotoxicity, sensitization, and systemic toxicity. The evaluation must represent the final, finished, and sterilized product. In vivo validation involves animal trials using approved models, such as alveolar bone defect. The study design includes test, commercial control, and blank control groups. Observation endpoints cover macro, micro, and cellular analyses over a minimum of 6 months. Sterilization assurance involves method selection and SAL validation, with strict warnings regarding EO gas and irradiation. Life cycle stability requires shelf life testing, including accelerated and real-time aging and transport stability validation. Clinical evaluation decision pathways offer the equivalence route, utilizing comparable existing data, or the clinical trial route, required for novel materials. Overseas clinical data may be acceptable if it meets NMPA technical guidelines. The NMPA submission blueprint relies on progressive validation, layering quality management, biocompatibility, non-clinical research, and in vivo clinical evidence, culminating in administrative and labeling documents. A structurally sound dossier moves from material identity to clinical efficacy. Thank you for watching this video. For more information, please reach out via email at info@whitneyconsulting.net or visit our website.